today we are talking about the structure of DNA. So, yesterday we talked about what DNA was made of, and some of the scientific experiments and scientists that were involved in making that discovery. Can somebody tell me one of the scientists that we talked about yesterday? Yes. Yes. Albert Hershey and Martha Chase. What'd they do? Yes, they did the experiment with the blenders and the viruses that helped determine that DNA was the genetic material. Anybody else have any of the scientists we talked about yesterday? About four. Give me one more. I'm gonna be asking you a lot more questions this year. Anybody? Any of the other scientists we talked about yesterday besides Hershey and Chase? Yes. Oswald Avery. He was one of the scientists that worked with the S and the R bacteria, the safe and the harmful bacteria that may or may not have caused a pneumonia in mice. So where we left off yesterday. Oh, it's not. Oh well. Okay. So where we left off yesterday, we want to know. We already know that DNA is the genetic material, thanks to Oswald Avery and Alfred Hershey and Martha Chase, and last name's escaping me. And we know what DNA is made of, thanks to Erwin Chargaff. We talked about adenine, thymine, guanine, and cytosine, those four nitrogenous bases. That make up DNA and are always in the same proportions. So adenine always goes with which one? A is with, yeah, thymine. Adenine and thymine are always paired together. Guanine and cytosine are always paired together. So we know that DNA is a genetic material, and we know what DNA is made of. But the whole goal of this, the entire biological community, is all about how DNA works. We want to know how this very, very simple molecule can lead to all of the genetic diversity on Earth. Every living thing has DNA as its genetic material. So if every living thing just has a bunch of A's, T's, G's, and C's, how on Earth does that give us everything from an earthworm to a human being? So that's the goal. But in order to understand how DNA works, we need to know what it's shaped like. All of these experiments that we've learned about so far have all been mostly chemical. We know what DNA is comprised of, and we can detect it in some other ways. We can't, especially not in the 1950s, look at it under a microscope. We don't have microscopes that powerful, at least not back then. So the race was on to find out the structure, the shape of DNA, the structure of DNA. That was the entire goal of biologists for a very, very long time. Two of the main characters in this race to figure it out is Watson and Crick, and I chucked Wilkins in there at the very end, and Rosalind Franklin. These are the two main characters in our race to discover the shape of DNA. James Watson was an American biologist. He actually got his master's degree from IU, which I'm not necessarily the biggest fan of him, but that's not the biggest reason I'm not super into that. And then Francis Crick was a British physicist. Um, and then Maurice Wilkins actually worked with Rosalind Franklin, but wound up in the Watson and Crick category. Um, and I will tell you all about that story. Rosalind Franklin was a British scientist. This was the mid 1950s, early 1960s. So, again, not a super long time ago. So, when you think about all that we know about DNA now, and 70 some odd years ago, we didn't even know what it was shaped like. So, we've come a very, very long way. And these people unlocked the door for all of the stuff we know now. Without these people, our understanding of genetics would be miles behind where it is now. So, James Watson, Francis Crick, Maurice Wilkins, and then Rosalind Franklin. I'm going to spend some time here just because I think this story needs to be told. The amount of people or the amount of times I've sat in a biology lecture and just heard about Watson, Crick, and Wilkins, and people just completely brushed over Rosalind Franklin's contribution is more than I'd like to count. So I want to make sure that you guys hear her story. So Rosalind Franklin was a biochemist. So all that stuff we learned at the very, very beginning of the year, how chemistry interacts with biology, right? All of those macromolecules. That's her field of study. And she worked with X-ray crystallography. What X-ray crystallography is she took DNA, because we knew how to isolate it, right? She took DNA and bombarded it with X-rays. And the atoms in these in the molecule of DNA diffract the X-rays, and that was captured on film, much like an X-ray is when you break a bone, right? They send an X-ray through your arm. If the X-ray hits a bone, it's bounced back, and that's why X-rays, your bones appear white, is because that's measuring the X-rays that are returned. And the X-rays can just pass through the soft flesh in your arm, so that's why your skin and stuff looks not so clear on an X-ray. The same thing works with DNA, except it's on a molecular level. So it took a lot more practice. She took a lot of these photos. She worked very, very hard to get these photos until eventually she got this right here. It's a very famous image. It is photo 51. So she tried this 51 times and didn't get any decent results until this one. Photo 51 suggested that DNA was a helix. So if you think about a ladder and twist it, helix. That consisted of two strands that are a regular, consistent width apart. So it doesn't. So if it's a ladder, it doesn't go in and out. Now, if you look at a DNA molecule, I've got this one right here. If you look at a DNA molecule, that doesn't necessarily look like this. But if you took this and turned it this way, you see where we're kind of getting the idea? We've got our center bit right here, and we've got our outline around the outside. So all these little red beads would be this darker part right here, and we've got our rungs, and that's our base pairs. We'll talk about that later. But this image is the reason we know that DNA is shaped like this. Before this, we had no clue. We had ideas. We knew that Chargaff's law or Chargaff's base pairing rules suggested that we had something working in pairs, but we didn't necessarily know how those pairs interacted with one another. So that's Rosalind Franklin. That is what she did. Doing okay so far? Anybody have any questions? Just like my favorite scientists are talking. Okay. Moving on to Watson, Crick, and Wilkins. Ooh. I had pictures. Whoops. Okay. So Watson and Crick worked together to try to figure out how DNA was shaped. And instead of doing something with biochemistry, their main goal was to just make 3D models. If we go back to this page right here, we've got this famous picture of them with their giant 3D model of DNA. They were just trying any physical confirmation where it would make sense that we had these base pairs, and how on earth those base pairs would be stored. Um, so that is Watson and Crick. But here's the thing: they weren't necessarily. I mean, they were intelligent people, obviously, but they weren't necessarily super close to a breakthrough until Franklin's colleague Maurice Wilkins got involved. So Rosalind Franklin had a coworker. His name was Maurice Wilkins, and they didn't necessarily get along. I mean, y'all probably know there are people you interact with at work or at school that you don't necessarily vibe with. So Wilkins worked with Rosalind Franklin, but he also knew Watson and Crick. And one day, while they were discussing the shape of DNA, Maurice Wilkins showed Watson and Crick photo 51 without Rosalind Franklin's permission. So he shared her findings with Watson and Crick without her knowledge. And based on photo 51, there is actually like quotes from James Watson saying that they weren't on the right track until they saw this photo. And they were able, from this picture, to figure out that DNA shape was this double helix. And I say twisted ladder. I don't know if this will twist it up or anything. But if you... okay. You see how that's kind of like a ladder? And as you twist it, you get that kind of DNA shape. Maybe so. So, Maurice Wilkins showed Watson and Crick Rosalind Franklin's work without Rosalind Franklin's permission. And Watson and Crick wrote about it. They had this proposed model that they built, this twisted ladder, and they published their findings in the journal Nature. Nature is a very, very respected biological findings journal. And so did Franklin. She published her findings, which were just photo 51, without all the flashy models, around the same time. But only one team got the credit. So Watson, Crick, and Wilkins won the Nobel Prize in Physiology or Medicine in 1953 for the discovery of DNA's shape, and they did not mention Rosalind Franklin. In fact, many people didn't even know about Rosalind Franklin's contribution until many years later. By the time that people figured out Rosalind Franklin was a major player in this discovery, she had passed away because X-rays are a carcinogen. If you work too much with X-rays, it can cause cancer, and she passed away of cervical cancer 5 years after the Nobel Prize was awarded. by the time people realized that she had this major contribution to the game, um, she was no longer around to accept the credit. And the Nobel Prize can't be given to people who have died. Uh, so she died not knowing that she would ever get the credit for what she dedicated her life to, literally. Um, because all of the work that she did or most likely is what caused cervical cancer and eventually. Do they have any questions? I think it's really important to tell her story just because it didn't get told for a very long time. Uh, I think James Watson's still alive. Not doing any scientifically credible stuff. He kind of ran into some scandals a couple years back, but yeah, no. So that is how the world discovered the shape of DNA. How we doing? All right. So now we know a heck of a lot more about DNA than Photo 51 and the fact that it was a double helix. We know specifically what DNA is made up of. So, if you think way back to our macromolecules, I don't know why I can't say that word. So when we talk about macromolecules, we talk about polymers, right? Polymers are things made of monomers. So our proteins, right? Our proteins are polymers, and they are made up of amino acids, which are their monomers. So DNA is a polymer, and its monomer is called a nucleotide. A nucleotide is made up of three things. It is very important you know these three things. So each nucleotide has a nitrogenous base, so either adenine, guanine, thymine, or cytosine. And if you remember from yesterday, it can be a double ring or it could be a single ring. So this would have to be one of our purines, just as this example. So it has a nitrogenous base, a pentose sugar. Pentose just means 5. So it's got 5 carbons in it. This is also known as a deoxyribose sugar, which is why DNA is called deoxyribonucleic acid. It's a nucleic acid where the sugar is deoxyribose. And then it has a phosphate group. So a phosphate group, a sugar, and a nucleic acid. If you popped off one of the nucleotides from my model, this little yellow piece is our phosphate group. This black piece right here is our pentose sugar, and this colored piece is our nucleic acid, or not our nucleic acid, our nitrogenous base. And they all fit together. I'll tell you how. They all fit together to form our DNA molecule. These three things are held together by covalent bonds. Actually, we're going to say, let's see. Our 3 parts of a nucleotide are held together by covalent bonds. And you don't have to write all of the like little numbers and letters in there, but it would probably behoove you to have some sort of picture, even if that picture is just a P connected to a pentagon connected to some sort of base. So this is a sugar, and this is our base. So our nucleotide, that's a pen. Is our phosphate connected to our sugar connected to our base. Okay, that's our monomer. DNA is made up of a bunch of these. Now, they're held together by covalent bonds because we need a very, very strong bond. We don't want to break these. If we broke these, that would be like all of these things coming apart. That doesn't do well for the structural integrity of our DNA molecule. So, those are our nucleotides. So, our nucleotides, if you look right here, so this is one, this is one, this is one, and this is one. They are held together by hydrogen. Yeah, hydrogen. So if you write this little sentence right here, the hydrogen bonds between nitrogenous bases hold our two halves together, you can simplify this picture by just going with our little pentagon, phosphate, base, base, phosphate, right? And this right here, this is what we want. This is our H bond. So if this is an A and this is a T, they are held together by hydrogen bonds. In my model up here, these little white pieces are our hydrogen bonds. Hydrogen bonds are weaker than covalent bonds. That's because we need access to split them apart. It's not that easy. But at some point in time, we will learn about how DNA replicates. We will learn how DNA is read. And when we do that, we need to break them apart at the middle and have two strands as opposed to one double helix. So these hydrogen bonds down the middle, they're much weaker than the covalent bonds because it's much more important for the backbone to stay in place than it is for the two halves to stick together. That make sense? So, like I said before, our general overview, DNA is a twisted ladder with complementary strands. Complementary because our A's are always going to bond with T's, and our G's are always going to bond with C or T. Yeah, C's. So if we had one strand of DNA here, and its complementary strand over here, this is A, T, G, C, C, G, A, T. The complementary strand would be T, A, C, G, G, C, T. Right. Does that make sense? Chargaff's rule, so him finding out that the percent of adenine was the same as the percent of thymine, and the percent of guanine was the same as the percentage of cytosine, was really, really important because we know that our two sides of our DNA molecule are going to complement one another. They're not identical, they complement. Right down the middle, these are hydrogen bonds. How are we doing so far? Okay. So, We know how DNA was found. We know how DNA structure was worked out. We know what the monomer of DNA is. We know how those pieces fit together. There's one more rather critical piece of information that if you get now will make protein synthesis a lot easier. So if you don't get this, let me know. And that is our strands are anti-parallel, meaning they run in different directions. So if you think about a pentagon, right? They're not necessarily symmetrical when it comes, or this way. So there's always going to be either an arrow pointing up or an arrow pointing down, chemically speaking. So one of these strands is going to have the sugars facing down. While the other strand is going to have them facing upward. Does everybody see what I mean when I talk about them pointing in different directions? Now, here's where it gets a little chemistry based. So whenever chemists look at one of these pentagons, one of these 5 carbon sugars, they number the carbons. And each one of these points represents a carbon molecule. And they number them 1, 2, 3, 4, and 5. Just like that. So when they're looking at the outside of a DNA molecule, they can see this one runs with the 3' on top and the 5' on the bottom. They say prime just to tell you that it's carbon. So it's going to go from 3 to 5 on this side. But if you look at this segment over here, we've got the 5' carbon on top, this one, on top, and the 3 carbon, this one, on the bottom. So if we're going from 3 to 5, it's going to run this way. We've got one of these sugars pointing upwards and one of these sugars pointing downwards. They're still parallel because they don't touch one another. But we say they are anti-parallel because they're pointing in different directions. Anybody have any questions here? On a scale of I'm completely lost hope and I got this, where are we? We'll do some more work with this one. So we've got our 2 strands of DNA, one's pointing upwards, one pointing downwards. They are anti-parallel. And that's the big takeaway, is the concept of them being pointed in different directions. The 3' and 5' was just to explain how we got there. So you don't have to worry about counting carbons, just that they're pointing in different directions. And when we read DNA, that's when it'll come in. Like I said before, base pairing, adenine is always bound with thymine, and guanine is always bound with cytosine. It's coming up a lot because if you get this now, you're going to be flying through the rest of this. ATGC. Actually, at some point in time, I think I grew out of it. I had a T-shirt that was the ACDC logo. That's the ATGC. I'm very proud of it. That tells you how big of a nerd I am. I have it somewhere. Anyway, so Base pairing, adenine's always with thymine, guanine's always with cytosine. Wherever there's an A, there's a T. Wherever there's a T, there's an A. Wherever there's a C, there's a G. Wherever there's a G, there's a C. I should be able to give you a strand of DNA like that and ask you to give me the other strand. So, G matches with, shout out, C, A, T, G, C, G, A, T. Beautiful. Get used to doing that. Every single strand of DNA, from the DNA in your bodies to the DNA in whatever it is you ate for breakfast this morning, if it was a plant or animal product, follows these rules. Every strand of DNA in every living thing that has ever lived on planet Earth follows these rules. They are made up of phosphate groups, sugars, adenine, thymine, guanine, and cytosine. Those 6 things makes all of the directions for all living things on Earth. Full stop. Period. The complicated simplicity of biology is one of my absolute favorite things in the world. So there is an exit ticket on Canvas. If you could fill that out for me, please. If you haven't filled out the one from yesterday, please take this time to do that as well. Those surveys really help me out. If you have any questions, comments, concerns, if you just want to talk about this stuff, like I said, I absolutely love it. So I am more than happy to spend more time with it, re-explain stuff, retell stuff, all that.